name is Salah Elbashir. Welcome on board and thank you for joining our bacteriology lab. I hope by the end of this lab you understand what's meant by gram stain and how it's used in bacteriology and why we use it. Also, I hope that you will enjoy this lab and understand all the aspects of the gram stain. Lynn, Mayar, and Faris will work with me and also with you and will help you to understand everything about gram stain. Gram stain is the most popular stain in bacteriology. It is a classical differential stain that classify bacteria into two big categories: gram-positive bacteria and gram-negative bacteria. As a differential stain, it requires 4 important reagents. And also, as a differential stain, those reagents should be used in order and applied to a heat-fixed bacterial smear. The 4 reagents, as listed by Mayar here, are the following. Number 1, the primary stain. In this case, it is a chemical dye called crystal violet. This stains the bacterial cells violet or purple, and this stain is considered a basic stain or a base stain that is actually charged positive. The second important reagent is called mordant, which in this case is gram iodine, and this reagent or the gram iodine serves not only as a killing agent but also as a mordant that intensifies and increases the cell affinity for stain, for the crystal violet stain. And it does this by binding the primary stain, forming an insoluble complex called crystal violet iodine complex. This resultant crystal violet iodine complex serves to intensify the color of the stain, and at this point all the bacterial cells are stained bluish or purplish black. The third one is a decolorizing agent, and this decolorizing agent is either ethyl alcohol with a concentration of 95% or might be an ethanol acetone. Both of them has a dual function, as it serves as a protein dehydrating agent and at the same time as a lipid solvent. Those two actions determined by 2 factors. Number 1 is the concentration of lipids in the cell wall, And number two is the thickness of the peptidoglycan layer in the bacterial cells. In the gram-negative and the gram-positive organism, they should have those two layers. The difference is about the proportion of each. In gram-negative bacteria, there is an abundance amount of lipids in the wall and a very thin layer of peptidoglycan that allows the decolorizing agent to push them post. And vice versa in case of the gram-positive bacteria, where it has a very little amount of the lipids compared to the gram-negative, and a very thick layer of peptidoglycan. And this makes a big difference when decolorizing, because alcohol cannot wash out all the peptidoglycan and thus does not allow the primary stain to be washed out. The last part of this, or the fourth reagent, is called the counter stain or the secondary stain. In this case, it is safranin, and it is a red stain. It stains the cells with reddish coloration. However, the most important thing to be known is that safranin is also opposing the crystal violet in its charge, as it is a negatively charged stain. That means it is an acid stain rather than the primary stain, which is a base stain. Thus, the idea behind using gram stain, and also the idea behind why gram stain is the foremost and most popular stain used in bacteriology, is that it is easily divide a big kingdom into two categories: the gram-positive stain bacteria and the gram-negative stained bacteria. Using chromophore classification, that means color classification. Second, using electrical affinity, electrostatic affinity of such bacterial cell to divide them into positive or negative bacteria. In this slide, I would like to emphasize that by differentiation, we mean that the stain divides bacteria into two categories based on their electrical charge affinity and the coloration appearance of the output after staining. The last but not least is that this differentiation depends mainly or principally on the chemical and physical characteristics of their bacterial cell wall. That is to say that based on the structure of the cell wall, it gives more opportunity to divide the big kingdom of bacteria into two main categories or sub-kingdoms. The first thing is that the layer of peptidoglycan, and the second is the amount of lipids contained in the cell wall. In case of a gram-negative bacteria, there is a thin layer of peptidoglycan and an abundance amount of lipids. This allows the decolorizing agent, which is ethanol, 95%, to wash out those two layers and lipids content, and then allow the primary iodine complex to be washed out, and then permits entrance of the counter stain, which is safranin, and this gives the reddish coloration of the cell. And vice versa in case of the gram-positive bacteria, where the peptidoglycan layer is large layer and very little amount of lipids in the cell wall of this type of bacteria, which does not allow the wash out of big amounts of lipids, and also the pores are very little, so the crystal violet iodine complex is very bigger, cannot be washed outside, and then the bacterial cell retains the purplish black color and become a purplish in color under microscope, and thus it is now a gram-positive bacteria. Is there any determinant factor? Yes, there are some determinant factors, and those determinant factors are as listed by Maillard again, decolorization, by Lean, decolorization of the primary dye, decolorization of bacteria, and the chemical and physical characteristics of the cell wall. Those are three determinant factors and are very important to be known. The procedure is very easy based on the FDA and their big manual, the Bacteriology analytical manual, is number one, that when you prepare your sample and have a stain heated or heat fixed films, then you just pour on it or flood it with crystal violet, and this for 1 minute, and then you wash with tap water. The second, you apply the grams iodine for 1 minute, then wash it with tap water again and drain it. And after that, just decolorize with 95% ethanol alcohol, and until blue color is no longer appeared, and then wash it with tap water again and drain. Lastly, you just apply safranin for around 30 seconds and drain, and also plot or leave it for air drying. Two things you have to understand: do not use too much of the crystal violet in the first place, because this will mask the whole thing. You can't make those steps very practical, and you can't see anything just blackish coloration, or even you can see the because of the much presence of the crystal violet, you can see even the negative bacteria as purplish. This is the most important thing you have to know. Also, you do not need to over decolorizing. You just need them for sometimes around once or twice washing is quite enough. By this, we reached the end of our lab session today. I hope it was enjoyable, and I hope that you understand all the aspects of gram stain and know why it is very popular. Next time we are going to see something else, but till then I hope I can see you well, all of you. Be careful about yourself and take care. If you enjoy and like this video, please give us a thumb. Try to follow us and prescribe in our channel, and try also to activate the bell, so everything you will be notified by any new video comes or we published. Thank you for listening.